The next speaker is Joy Buongiorno. Joy received her PhD from the University of Tennessee in Knoxville under the supervision of Karen Lloyd. She's currently a postdoctoral researcher at Carnegie, where she's working with Shauna Morrison and Bob Hazen. She will be talking about microbial communities and proteins associated with volcanic hot springs, hydrothermal vents.
So I have greatly enjoyed the talks both today and yesterday. A lot of great things to start thinking about, being updated on the different themes that I'm looking to collaborate with. I'm especially excited about the work that Ariel and Adrian are doing. So today what I'm going to share is just a quick update about the database that we've been doing under theme 3, the coevolution between microbial ancestors and the geosphere, specifically focused on tectonic settings and their control on oxidoreductase assemblages. And I want to point out straight away that this is definitely not a sole effort. This is a huge compilation of efforts across multiple institutions, including Donato Giovanelli, Shauna Morrison, our collaborators at RPI, who Bob introduced just one of them earlier, and then Marie Edmond, who we just got on board. She's going to help guide us in our questions that we begin to ask of this database. Okay, so as we've been talking for the past 2 days, it's all about understanding feedbacks between the biosphere and the geosphere. So Kara yesterday in her talk talked about whiffs of the oxygen that we saw potentially being attributed prior to the GOE to the rise of these stem groups of cyanobacteria. But really what the GOE represents is this massive change in planetary redox chemistry. And what that brings about is, of course, a whole suite of new geochemical reactions that then imparts differences in terms of the availability of trace elements. So we've begun to discuss this in terms of copper availability as well as molybdenum. And so those are things that we can clearly see from the rock record increase in their availability after the GOE. So the big question is, what does this mean for microbial communities? Well, we have all of these new bioinorganic elements that are available. This spawns a great host of metabolic diversity, and therefore there's other things to press feedbacks on the environment. Biological drawdown, metabolite generation, things of this sort. So this is the crux of ENIGMA in general. And really what I'm interested in is the sources of these trace elements in modern environments. This is a paper put out by Edmonds et al. In Nature Geoscience in 2018 that took a look at the fingerprints in trace metal concentrations between different volcanic settings. So we can see here in the blue, we have arc volcanic plumes. In the red, we have hotspot volcanic plumes. And just by and large, you can see that there are differences in these bioinorganic elements that we're interested in. So for example, we have more enrichment in molybdenum and arc volcanic plumes and copper as well. And this is important because as we have been talking about, these different enzyme cofactors can be incorporated and really impart different redox potentials on these oxidoreductases. This is the chemical reviews paper that was brought up at the beginning of the day by Vic, by Yilu and colleagues, and this just points to the differences that the metal centers can really make in terms of the functioning of the oxidoreductases. So we have two driving hypotheses. Number one, encoded oxidoreductases are constrained or influenced by the availability of trace metals in the environment. And number two, specifically different tectonic settings dictate trace metal availability. So the way we go about answering this question is called abductive science. It's not starting from a general premise, it's just collecting as much data as we possibly can to try to find emergent properties that maybe speak to these hypotheses in a stronger way. So walking through the steps to address these questions, this is the multi-dimensional analysis workflow. It starts with database mining with the tool that Adrian developed as a PhD student called MetaSeq. This allows you to search a query word like hot spring or hydrothermal vent. It then goes to the short read archive that's hosted by NCBI and pulls out any metagenome that matches that query. We then do short read annotation with MyPhaser, which Adrienne talked about in her talk as well. And this is just a rapid annotation tool that doesn't require metagenomic assembly, and it does some quality assessments on the front end too. After that, we do quality control and normalization of this abundance data that's given to us with the MyPhaser tool. We're starting to rethink how we do normalization right now of our data, so we're actually playing around with all of this at the moment. Once we have good data, we can start to play around with different correlation metrics, like the Spearman correlation. I've been using R to do this. We can also apply clustering and click analysis algorithms on that data. We can visualize our results and then identify drivers of those clusters of oxidoreductases or clusters of communities with different machine learning algorithms. I have been using random forest, but again, we're starting to play around with different algorithms. Okay, so the last time we spoke in April, I think I had a fewer metagenomes that we have right now. So our database that we just closed off is at 787 metagenomes. These represent 259 hot springs represented by orange. We've got deep vents and shallow vents, which are hydrothermal locations. We have geysers, fumaroles, and volcanic lakes. So we have a really great geographic swath represented in our database. But as with any good database, a lot of curation has to go into this. So that is the first step. And we can take a look at the composition of our database, and we see that we did a whole bunch of manual discovery efforts as well as metaseq efforts, and taking a look at how many unpublished metagenomes we have, so things that weren't available in the NCBI database. This represents about 40% of the unique biosamples that we have, and a biosample is just a way to reduce the amount of data that we have. So let's say we went to NCBI and one researcher sampled the heck out of one hydrothermal vent, and another researcher at another hydrothermal vent only had one sample for that vent. So in order to make those comparisons equitable, we have to do some data reduction. And overall, we mostly have fluids in our data set, but we also have some biofilms, rocks, and sediments. So in addition to the oxidoreductase and metagenomic side, we also are very, very interested in putting these microbial samples into a geological context, of course. And so we did a lot of manual cross-referencing in geological databases, as well as the literature for geochemical data. This is things like querying the MORB database, the Mid-Ocean Ridge Basalt database, plugging in some latitudes and longitudes for the metagenomes we have, and seeing what the chemical composition of that crust is. EarthChem Portal is another beautiful tool that lets you query based on latitude and longitude, or you can create polygons. So that's been super helpful as well. Also specifically, there are RCNs for specific sites. The Yellowstone National Park has their own, and this was very helpful in determining some of the dissolved trace metal compositions for some of the sites that we didn't have access to the geochemistry otherwise. In addition to aqueous geochemistry, we also want to put things, of course, into a tectonic setting. And this is a beautiful schematic put together by Donato Giovanelli, just taking a look at the different types of tectonic settings in a hierarchical fashion. So tectonic settings can be either characterized as plate margins or interplate. Within the plate margin, you can be a convergent margin where things push together and you have a subduction of one slab over another slab. You could be a divergent margin where you have a plume of magma pushing up, creating a spreading center here. You could have transform margins and also passive margins as well. Intraplate, we have rifting, but we also have hotspots. And the most notable hotspot that you might be familiar with is Yellowstone National Park. And so in our database, we have these different types of tectonic settings represented. Taking a look at the different features that we can count in our metagenome. So along the x-axis here is metagenome count, going all the way up to the 787 non-unique samples that we have. And taking a look at the chemistry that we have, both from EarthChem as well as the MORB database, we see a lot of our metagenomes have data for iron and manganese, not so much for molybdenum. But what's really cool is we just got this whole suite of data from Sabin with G plates, looking at different parameters that characterize the tectonic setting itself. So we have things like plate velocities, heat flow, elevation, things like slab and dip, things that you may not think may matter so much to the microbes living on the surface or at the sediment water interface, but it might matter. Yeah, sediment water. We can begin to query with more traditional geochemistry metrics, the chemistry that we do have. So this is a ternary diagram with each apex of the triangle representing 100%, 100 relative percent of either aluminum, magnesium plus iron, or sodium, calcium, and potassium. So looking across the different types of samples we see that we have different chemistries characterizing these samples, which is great. We do want variety and diversity in the samples that we're exploring, just to maximize the potential to see differences. Here's just a typical pH and temperature diagram with location indicating the color. So we see across the samples, we have things that are characterized by pretty low temperature, but very high pH, as well as the opposite, where we have a low pH environment and a pretty high temperature. So again, this just speaks to the variety of geochemistry and geochemical regimes that we're dealing with in our data set. Okay, so moving on from curation and what we have, let's take a look at MyPhaser. And so the first thing that microbial ecologists like to look at is diversity. So I wanted to know, are oxidoreductases being well represented in our data set? So we can take a look at alpha diversity in a simple accumulation curve here with metagenomes, number of metagenomes along the x-axis, and the number of oxidoreductases that are in the metagenomes along the y. And we see that overall, the 320 oxidoreductases that are in the MyPhaser database are pretty well represented in our metagenomes. We can take a look at a different metric of diversity called the Shannon Index. And the Shannon Index is a measure of richness and evenness in the samples. And here I've separated on the left different hot spring samples that we have. So each dot is a specific metagenome. And on the right, we have hydrothermal vents. And I've colored these by the tectonic settings, again, looking at the hierarchy. So are you an interplate? Are you a plate margin? And so what we notice is that we have high oxidoreductase diversity observed for many of the locations here. But we also see the largest range is observed at the continental hotspots that we have. These are largely samples from Yellowstone National Park. So the next question is, how are oxidoreductases distributed across all of our samples? We can answer this question by looking at community networks, which we've been presented several different types of networks today, and I'm going to give you another one. So this is a community network, and what I mean by that is that each node represents a metagenome, and it is capturing the abundance of a suite of 256 oxidoreductases. The 256 oxidoreductases represent the most abundant oxidoreductases within the samples. So again, we are coloring the nodes by the different tectonic setting. And first, we notice that the topology suggests distinct groups of communities. So here at the top in red, we have hotspot samples. They've kind of split off from each other. And we also have hydrothermal vents balling up into this one little hairball in the middle. So what we can do on top of just visualizing these communities is we can ask, are these communities able to be discerned with different clustering or click algorithms? And so what I did is I used the Louvain module detection selection tool in R to identify clicks or clusters or communities. Everybody has their pet word for it. But the next slide is going to be the same network, but instead of the node being colored by tectonic setting, it will be colored by a click. Here we go. So what we see is that I'm just going to go back. We do, in fact, have different communities of our hotspots. So what was all red over here is now green and orange. And then here in the middle in this hairball, we have 2 distinct hydrothermal vent communities here. So we can ask, what are the compositions of these clicks? And we see that we have continental hotspot hot springs representing click 1. These are these green nodes over here. So all of these samples come from the same type of tectonic setting. Click number 2 is represented in pink over here. And most of these are divergent margins, as well as the composition of click 3 as well, represented in blue that you can see. The hot springs that are located in click 3, though, are different settings compared to those in click 1. And then finally, this last click seems to be like a grab bag. These are mixed with plate margin and intraplate, oceanic and continental, as well as a mix of reduced and oxidized regimes. So I should say this work is preliminary. We just closed the database. We're now beginning to renormalize our data. And once that is complete, I'm going to go back and generate these networks again and make sure that these results are robust. In addition, I want to analyze unique biological samples only. So if you notice this hairball, that's the problem that I spoke of earlier, where you have research teams that sample one site over and over and over again. And so you have an overabundance of one site in your data. So we're going to subtract and do some data reduction to get rid of that problem. We also need to start thinking about batch effects as well. So does the team of researchers and their methods for DNA extraction and sequencing and amplification, does that matter? So preliminary conclusions, the database that we have is a good representation of tectonic settings. I should also point out that in addition to the many features that I've already shown you, we have many, many more. I lost count last time I looked at the database, but we also have a whole swath of mineralogical assemblages. So if that data will be of use for any of your projects, please let me know. We're happy to share this database once everything is finalized and ready for people to start playing around. And the results suggest that different oxidoreductases are at play at different tectonic settings. Hotspots show the greatest range of diversity in the oxidoreductases that are in the MyPhaser database. Network and click analysis is able to distinguish between hydrothermal vent metagenomes and the terrestrial hot spring metagenomes that we examined. We observed different groupings of oxidoreductases from hot springs within different tectonic settings with the click analysis that we did. Things that are ongoing and upcoming, there's a lot. It's a mad dash to get this done. I start a new faculty position at Maryville College on August the 1st. So I want to get this wrapped up as much as I can with all of your help. So this is where this comes in. We need to finalize the normalization and circulate this database to members of the ENIGMA team and others who might benefit. And I want everybody to take their collective knowledge, their background knowledge, and whatever interests them, and look at different aspects of the geochemistry, the enzyme composition, or the subduction parameters. Just attack it at whatever angle you do in your own data sets. And so hopefully the marriage of that knowledge with what we're coming at as far as driving questions will really make a beautiful paper at the end. We're hoping to collect the results from everybody's efforts in the form of some sort of informal seminar meeting. Donato's spoken about this, and so is Shauna. So we need to figure out the timing of this and when that could all happen. So please stay tuned. And we want to incorporate, of course, these findings in the current draft of the manuscript that's being written right now. So the future, there's always more things to do. Expand this type of data analysis and collection to include other environmental settings that are not necessarily tectonically active, like sedimentary basins or metamorphic provinces. And then Ariel from Jana's lab have been developing this tool called MyMetal. I don't know if the name has changed, but we're also interested in the distribution of metal binding proteins across these different tectonic regimes. So marrying his tool with our data in the end, I think could present a really beautiful product. All right, so with that, if there's time, I'm not sure, I will take your questions. Maybe I can turn my camera on.
Thank you, Joy.
Nathan, can I ask a question?
Yes, go ahead.
So what's going on in hot springs? Why is there diversity of oxidoreductases there?
I think it's just the noted case of each hot spring is such a mesocosm in and of itself. It's like a natural laboratory. And so I don't know what the species composition looks like in these places, but I imagine that the communities might be very different. There might be one community that drives the diversity and it's totally like dominated by one thing. But yeah, that's the only thing I can think of.
So just to clarify, the diversity is when you're sampling across multiple hot springs, not huge diversity within a particular location.
Right. Do you mean like within a simple hot spring?
If you were to take one specific hot spring, is the diversity of oxidoreductases high there, or is it because every hot spring is unique in terms of its chemistry?
I think it's unique in terms of its geochemistry and its microbial inhabitants. Yeah, it could be very high or it could be very low. It's a very wide range when we're taking a look at the Shannon Index just as one measure compared to the hydrothermal vent samples that we're seeing.
Saroj, I guess, has worked a lot on that. If you go to Yellowstone, there are hundreds of hot springs, and every one of them is different in terms of chemistry. So the microbiology of each hot spring is quite unique. But I want to ask you another question. So Mike Russell and others have invoked for many years hydrothermal vents as a source of the origin of life because of the serpentinization reaction, being a production of hydrogen. Hot springs have another possibility here. And because, I mean, I'm not sure where life began. Obviously, none of us really know. But it seems like there were probably many more hot springs than hydrothermal vents, although I'm not sure if I know that very well. Jiwa probably knows that, or Bob, in the late Hadean. Any thoughts?
I don't know what I did with my, there we go. So you're asking what is more likely for the origin of life?
I mean, you know.
Yeah, they had to contend with a lot on the surface. So that probably wasn't necessarily the case submerged. And I think it was a couple months ago, somebody released a paper making the case again for a warm little pond being the origin of life. But yeah.
That was the letter of Darwin wrote. But I mean, you know, both hot springs and hydrothermal vents are relatively short-lived. Tens of thousands of years, maybe, not millions. So it's an interesting problem. If life started in a hot spring and it couldn't move out to someplace else pretty quickly meaning thousands of years, then the experiment would have been over.
Yeah, I think that's an argument against like the small little pond, but I think an argument for the small little pond, as Doug is polymerization reactions, you know, often, I mean, depend on condensation reactions, which in wetting and drying environments could be, you know, more frequent. So if you wanted to make something bigger than a couple amino acids long, for instance, a pond might be more likely than a hydrothermal vent, possibly.